Hey everyone, in this video I am going to talk about a technique called massively parallel sequencing. And there are some other names for this technique, so also known as second generation sequencing or next generation sequencing. And we can contrast massively parallel sequencing with what we just covered recently, which is Sanger sequencing. So another name for Sanger sequencing, now that we have these new sequencing technologies, is first generation sequencing. So with massively parallel sequencing, we can sequence millions, perhaps even billions of DNA molecules at the same time. And these, all the technologies, there's several different technologies that can perform massively parallel sequencing, but one of them uses what we will call a slide. So it's like a microscope slide shaped thing that we can use for sequencing, and the slide surface is very important for sequencing. Over here with Sanger sequencing, we learned about how most Sanger sequencing nowadays is done with capillary tubes. And so with this slide-based process, we can put like millions to billions of molecules of DNA on this slide. But the capillary tube, that's one molecule per capillary tube. And I think there's somewhat high throughput ways you can increase the number of sequences you can do at the same time with Sanger sequencing, the number of DNA molecules you can sequence just by adding more capillary tubes. So, but however, no way is it going to reach the millions to billions of sequences at the same time. So this massively parallel sequencing, these technologies are really major breakthroughs when we are talking about genetics and molecular biology. So there are a number of different technologies that can perform massively parallel sequencing, and we are going to focus on what is currently the most popular technology called Illumina technology. Now, Illumina is the name of the company that the company that uses the technology that we will be describing today, or I will be discussing in this video. So massively parallel sequencing also goes by the name second generation sequencing. So essentially, we can use second generation sequencing technologies to sequence, let's say, a whole genome. So all the DNA, the DNA in a genome. That might be all the chromosomes in the nucleus, might include the mitochondrial chromosome, if we're talking about human cells. So we can sequence all of the chromosomes. Now we can also use this technology to sequence, let's say, all of the total RNA. So all the RNA in a cell, or a collection of cells, we can sequence all of those RNA molecules if we want to. We can sequence specifically the messenger RNAs. So instead of all the RNA, well, maybe we want to know what all the sequences of all the messenger RNAs that exist in those cells. Well, we can use second generation sequencing technologies to do that. We can also, instead of messenger RNAs, we could sequence all the small RNAs. We can also adapt second generation sequencing technologies to do all sorts of interesting research related things. So for example, we could use this technology to ID all of the methylated regions of a genome. So we'll talk more about DNA methylation in the future, but some parts of the genome are methylated. The DNA is methylated for various reasons. And if you want to identify those regions of a genome in a certain organism that are methylated, well, you can use this type of technology. You can also use this type of technology to identify, let's say, DNA-DNA interactions. And why would you want to do that? So let's say you are studying how DNA or chromosomes are organized in a cell. So you can use this technology to say, okay, well, these parts of chromosome 3 are next to these parts of chromosome 5 within the nucleus. And not just those specific parts, but you can see what parts of the genome are next to what other parts of the genome using this type of technology. You could also use it, let's say you have a protein and it's a DNA binding protein and it's bound to, it binds somewhere in the genome. You can use this technology to figure out where that protein binds, what sequences this protein binds to. And maybe there are many sequences. You can use this technology to figure out all of the sequences that the protein binds to. So let's say DNA or parts of genome, parts of a genome bound by a protein. So there are a lot of things that you can do with this technology, and throughout this course we're going to see many instances of researchers using this technology to identify important things about molecular biology. So all of those methods require what is called the preparation of a DNA library, even if you are, or DNA sequencing library. So let me talk a little bit about what a DNA library is. So DNA library essentially is this term. When we say a DNA library, what we mean is just a collection of DNA molecules. So the first step to massively parallel sequencing, the first step to second generation sequencing is to make what we call a DNA sequencing library. So essentially what that means is taking all of the things you want to sequence, all the nucleic acids, whether that's DNA or RNA. If it's DNA, well, you're just going to turn that DNA you're going to prep it in a way so that you can load it into the sequencer. If it's RNA, well, then you need to use some sort of reverse transcription to turn it into DNA to sequence that with the sequencer. So the first step is going to make a DNA sequencing library that can then be loaded into the sequencing machine. So there are a number of different ways to make a DNA sequencing library for use with, let's say, Illumina technology. The one we are going to go over in this class is called the NEB, for New England Biolabs Next DNA library, or DNA sequencing library preparation method. We're going to go over this one. And if you can understand this one, well, essentially you can understand all the different methods there are to preparing a DNA sequencing library for use with Illumina, or for sequencing with Illumina technology. Okay, so what we're going to do is, overall, our goal is to learn how Illumina second generation sequencing works. We are going to focus on the NEB Next DNA sequencing library prep method. In these videos. So, I should say in this video. So what we're going to do, the first step is to get DNA from the organism. Now this is something I haven't talked much about yet this semester, like how do we get DNA? There are special kits that are designed by biotechnology companies that make the process of isolating DNA from tissue relatively easy. So let's just start with, we're going to get DNA from an organism. And once we have that DNA, we can start preparing the library. Now, to prepare the library for sequencing, there are several steps. So first step is going to be DNA fragmentation. Second step is end repair. Third step is A-tailing. Fourth step is adapter ligation. And with the NEB next kit, there's going to be a step called U-excision. Size selection. And then amplification. Okay. So in this video, we're going to go over these first 4 steps, and I'm going to save these 2 for class. So for DNA fragmentation, okay, what is that? So there are a number of ways to do this, but let's say we have this DNA molecule here, and this DNA molecule here represents all of the DNA in our DNA sample. So all of the DNA from all the different chromosomes, let's say, and say we're sequencing a human genome. So humans, we isolated DNA from human cells, and we have all the DNA from chromosomes. And this DNA is tens of thousands to hundreds of thousands of base pairs long. Because, you know, when you're isolating DNA from cells, it's really hard to keep the DNA from breaking into smaller fragments. So let's assume that after we isolate DNA, the DNA is tens of thousands to hundreds of thousands of base pairs long. But for these sequencing technologies, what we need to do is we need to break them into smaller pieces. So we need to create DNA fragments of a certain size range. So in our example, we are going to assume that we want a size range of 400 to 600 base pairs. So we want all, or 90% of the fragments to be within a range of 400 to 600 base pairs. So we're going to fragment the DNA, and one of the most popular ways to do that is to use a method called sonication. Now that uses high frequency sound waves to break the DNA into smaller fragments. So we can use sonication, and there are ways to figure out, you know, how long or how long you should expose the DNA to these high frequency sound waves. So let's just assume we figure out the right amount of time, and we check the DNA, and we find that, oh yeah, okay, most of it is in our preferred range of 400 to 600 base pairs. 400 to 600 base pairs. So the problem here now is that we're breaking the DNA, we're physically breaking the DNA with sound waves, and we might have these jagged ends now with various 5' and 3' overhangs. Okay, and that's going to be bad for the next step. So we're going to need to fix that. And that's what the next step is all about. So the next step is called end repair. So step 2, end repair. So, okay, so what is end repair? Well, how are we going to do it? So one thing I want to point out is sometimes we will get lucky. Some of these fragments, and there are going to be millions of these fragments of 400 to 600 base pairs long. And what I'm doing here is I'm diagramming just a little DNA molecule, and it's not to scale, right? This is, I mean, it's not the right length, right? So this is representing a 400 to 600 base pair molecule. And we have our 5' ends and our 3' ends. And let's assume the 5' ends have phosphate groups and the 3' ends have hydroxyl groups. And well, this would be perfect. If all of the molecules looked like this, then we wouldn't need to do anything. We wouldn't need an end repair process. However, a lot of them are not going to look like this. In fact, probably most of them will not look like this. So one possibility I'll have this DNA molecule down here, and I won't diagram the whole thing. I'll use these little circles to indicate that it's a really long DNA molecule. But let's say one of the ends looks something like this, with the 5' end overhanging the 3' end. Now this is a 5' overhang. The 5' end overhangs the 3' end. Now another possibility is the 3' end overhangs the 5' end. And I just made up some bases here. So this is a 3' overhang. Okay, so these are problems. We need to repair the ends so they look like this. Now I didn't show the other ends over here, but we can assume that, you know, most likely they're probably jagged. They're probably jagged like this, or there is a good chance that they are jagged and they need to be repaired also. So they must be fixed. They must be what we would call blunt-ended. So this is where we have to review a little bit. So maybe, and I can take a break and sharpen my pencil. So maybe some of you remember DNA polymerase 1 from genetics or microbiology, or a course from, or maybe even introductory biology. So DNA polymerase 1, okay, so this is the polymerase from E. coli, and it has several different activities which are going to be important for end repair. So we're going to use this enzyme, but we need to review its activities before we understand why it's used and how it works. So one of the activities of DNA polymerase 1 is a 5' to 3' polymerase activity. Another one is 3' to 5' exonuclease activity. And the final major activity associated with DNA polymerase 1 is 5' to 3' exonuclease activity. So, and maybe you remember, I can refresh your memory by diagramming something like this. So way back when you were learning about DNA replication, and you had a replication fork here, and this is going to be in your previous classes, is you had your leading strand and lagging strand synthesis, you have a replication fork. Well, DNA polymerase 1, it's usually the one that we say is playing a big role in lagging strand synthesis, or at least filling in the nicks and gaps left in lagging strand synthesis. So maybe that helps you helps remind you of where you probably heard of DNA polymerase 1 before. So, but we need to review those things. So 5' to 3' DNA polymerase activity. So what does that mean? So it's just, you know, that's the standard activity that is found with DNA polymerases, right? So let's say this strand down at the bottom is our template strand. That's the 3' end, that's the 5' end. Well, DNA polymerase synthesizes DNA, or polymerizes DNA in the 5' to 3' direction. So if this polymerase is synthesizing DNA, it's doing so in the 5' to 3' direction with respect, that direction is with respect to the strand undergoing synthesis. So that's the 5' to 3' polymerase activity. Now, the 3' to 5' exonuclease activity. Okay, so I'm going to demonstrate that with this little diagram here. I probably don't need to diagram all these nucleotides down there, but I will. Okay, so let's assume that something happened during replication here. Something happened, or during polymerization here. And okay, some wrong bases, or one wrong base, even one wrong nucleotide was incorporated here. Well, what DNA pol 1 can do is it can use its 3' to 5' exonuclease to cut these out, going from the 3' end from the 3' end to the 5' direction. So going in the 3' to 5' direction, it can cut out these nucleotides one at a time. So that's the 3' to 5' exonuclease activity. Okay, now this last one down here, what is this? So this is useful during DNA replication, when DNA polymerase 1 is synthesizing DNA, it's going this way, and there's let's say an RNA primer in its way. And what it wants to do is remove that. What is it going to do? It's going to use its 5' to 3' exonuclease activity to get rid of these nucleotides so it can keep synthesizing DNA in the 5' to 3' direction. So this would be the 5' end here, and it's going to use its 5' to 3' exonuclease to chew away these nucleotides. So that's the 5' to 3' exonuclease activity. Okay. Still in frame here. Yep, still recording, I hope. Okay. So now some other interesting things about DNA polymerase that we are going to need to understand is that DNA polymerase 1, if we could diagram it, we can sort of, say that different parts of the protein are associated with the different activities. So let's say this is the normal DNA polymerase 1 over here. Now I can say the 5' to 3' polymerase activity, let's say, is associated with this part over here. And let's say the 5' to 3' exonuclease activity is associated with this part down here. And then that leaves us 3' to 5' exonuclease associated with this part right here. So researchers found a long time ago that, and don't quote me on this, I know it's a protease, and I'm pretty sure the name of the protease is subtilissin. So maybe I'll add a correction to the video if that is incorrect. But the subtilissin protease, what it does, it can cleave DNA polymerase 1 in such a way that it removes the 5' to 3' exonuclease activity. So that is removed, and leaving us with what is called the Klenau fragment. And the Klenau fragment can be separated. The Klenau fragment is larger than this other part down here, the non-Klenau fragment. I don't know why it doesn't get a name. Non-Klenau fragment. Or maybe it does have a name and I don't know it. So Klenau fragment is the big fragment left over after subtilissin protease treatment of DNA polymerase 1. And so the Klenau fragment retains the 3' to 5' exonuclease activity and the 5' to 3' polymerase activity. Okay, so if you ever hear a researcher saying, "Oh, we use the Klenau fragment," now you know. It's not just DNA polymerase 1. It's specific parts. It's a specific part of DNA polymerase 1 that has lost the 5' to 3' exonuclease activity. And there might be some molecular biology techniques or research approaches that want to use the Klenau fragment because they don't want it to have this 5' to 3' exonuclease activity. Maybe that activity would interfere. It could interfere with let's say, the goals of the experiment. So one more thing, actually, I can introduce this now. This would be a good time to do that. Is there is a version of the Klenau fragment called Klenau XO-. Now, I wish there was another protease that could just kind of get rid of this, but the Klenau XO- is just a mutant version of DNA polymerase 1 where this is no longer active. So essentially, if you get the Klenau fragment from a DNA polymerase that doesn't have a functional 3' to 5' exonuclease activity because of a mutation, well, then essentially we could say it looks something like this. So just the 5' to 3' polymerase activity. In reality, this other part is there, it just doesn't work. It just doesn't work. Okay, so there's going to be a part later on where we need to, or where a researcher would need to use Clan-Out XO- for making the sequencing library. So we're going to need both of these at different times. Okay, so back to, now that we've reviewed a little bit about DNA polymerase I and learned what the Clan-Out fragment is. So we have end repair of our DNA fragments that we were talking about. So one thing that the Clan-Out fragment will do for us, so we'll add the Clan-Out fragment to our sonicated DNA molecules. And what it will do is we're adding DNTPs also, right, because it's going to need some nucleotides. Let's say for the 5' overhangs, what it's going to do is use that 3' end to fill in that molecule using its 5', that's a terrible 5' to 3' polymerase activity to fill in 5' overhangs. So those 5' overhangs, it uses that 3' end, uses the 5' overhang as a template, uses the 5' to 3' polymerase activity to fill in the 5' overhangs. Okay, so now we have a nice blunt-ended fragment. All right, okay, thank you, Clan-Out polymerase. And it's not done. So here's our 3' overhang, another molecule with a 3' overhang, and it could be even shorter than that. So what Clan-Out fragment will do there's our Clan-Out fragment. It's an ugly Clan-Out fragment, sorry about that, but I'm showing that it lacks the 5' to 3' exonuclease. It's going to use its 3' to 5' exonuclease activity to chop away, or to eat away, this 3' overhang. So 3' to 5' exonuclease activity. What's it going to do? It's going to remove 3' overhangs. All right. Okay, so thank you, Clan-Out. Okay, it end repaired it. It end repaired our DNA fragments for us. So, unfortunately, we're not quite finished. We're not quite finished. So, what we're going to need to do, let's call this end repair part 2. So, ideally, all of our DNA molecules in the library at this point are going to look something like this, with no overhangs. No overhangs. However, we need one more thing. In order to go on to the next step, we need the 5' ends to have a phosphate group and the 3' ends to have hydroxyl groups. So the nucleotides at the 5' ends have to have a 5' phosphate, and the nucleotides at the very 3' end need to end with a 3' hydroxyl group. And so in order to ensure that this is the case, what we do is we will add an enzyme called T4 polynucleotide kinase. Okay, what does this enzyme do? So it adds phosphates to 5' carbons of 5' nucleotides. So the nucleotide at the 5' end, say over here and over here, if it doesn't have a 5' phosphate or phosphate group on that 5' carbon, it will be added. And what does it also do? So it also removes phosphate groups from 3' carbons of 3' nucleotides. So at the very 3' ends, if there's a phosphate group on there, for some reason, if there's a phosphate group on that 3' carbon, that last nucleotide, the 3' carbon of that last nucleotide at each 3' end, it will remove it. And that's important for the next step. Okay, so how can there be no, I mean, you probably have never even thought about a phosphate group on a 3' carbon. How can that happen? Well, it could happen that, you know, we use sonication to make our DNA fragments, and it could be that a large portion of them, you know, they look pretty good, they're blunt-ended, and they just don't have the phosphates and hydroxyls in the wrong spot. So let's take this 3' end right here. Maybe it looks something like this. So here's our nucleotide. You know, one of the structures that we definitely need to memorize the structure of a nucleotide. So here's our 5 carbon sugar, the base, the phosphodiester group, or part of it, that's going to be linking to this other nucleotide over here. So instead of an OH, you know, maybe during sonication, the bond phosphodiester linkage broke in a way that, you know, this oxygen is no longer connected to the downstream nucleotide. Okay, so it broke here instead of here. Okay, so we have a phosphate group on that 3' end. So that's possible. Now, we could also look at this 5' end. How come there's no, let's say there's no phosphate group here. Well, for that 5' end, well, maybe the DNA fragmented in such a way. So I'll diagram this phosphodiester linkage there. Now, let's see, here's our base. Here's the other base. Now, if this is the 5' end, this one right here is the next nucleotide downstream. Now, it could have broken in such a way that the phosphate group that was, you know, linking wherever this nucleotide was located in the genome to the upstream nucleotide, it broke in such a way that it removed the phosphate group. Oh, yeah, maybe it's this one. Maybe it's this one down here, right? And what we are left with is our 5' carbon right here. We're left with a hydroxyl. Okay, we need a phosphate group on that for the next step. Okay, so T4 polynucleotide kinase is going to take all these fragments right here, blunt-ended fragments, remove any phosphates that happen to be on the 3' ends, on the 3' carbons of the 3' nucleotides, and add phosphates to the 5' carbons of the 5' nucleotides. Okay, so that's end repair. Now we're ready for the next step. A-tailing. Okay, A-tailing, or adding A tails, sometimes called the DA tails. So what does that mean? So let's take one imaginary fragment that now represents all of our molecules in the DNA sequencing library. It's not finished yet, but all the molecules that we're trying to make into a DNA sequencing library. So millions of molecules, all of which have a various 400 to 600 base pair interval of the human genome we're trying to sequence. Now, ideally they all look like this. Okay, so now what we're going to do is we're going to use an enzyme that we already talked about, Clenau, XO minus. So this clanO XO minus, what does it have? The only activity it has left is the 5' to 3' polymerase activity. Now there's something interesting about DNA polymerase 1 that you've probably never heard before, is what it does when it gets to the end of a DNA molecule and there's no more template left, or even if it, there are just DNA molecules there, it'll bind to the DNA, but there's no template, so it doesn't synthesize DNA in the normal way. But what it will do is it will add an A nucleotide. So an A nucleotide to the 3' ends. So, and these will end with OH groups on their 3' carbons. So these are just regular adenine nucleotides added to the 3' ends in a template independent manner by DNA polymerase 1. Now the clanO fragment, because it doesn't have its 3' to 5' exonuclease activity, this version of it doesn't, it won't remove it. If it had it, what would it do? It would put the A on, take it off, put the A on, take it off. So by using this version of clanO, the XO minus version, it just puts these little A tails as one A nucleotide to the 3' ends of the DNA molecules. So at this point in the library preparation method, all of our DNA molecules, or most of them, ideally the large majority of them, should look something like this. 400 to 600 base pairs long with A tails, we call these, adenine tails, and 5' phosphate groups. Now, next step is going to be adapter ligation and U excision. So I'm probably going to stop this video here and start a new one just in case something is going wrong with the video, so I don't have to redo the whole thing from scratch. Next part, the next step of library preparation is going to be adapter ligation and U excision. Now this U excision is specific to the NEB Next library preparation method. Now, why do we need, what is this adapter ligation? Okay, so all of our DNA molecules, essentially they're just parts of a genome that have been end repaired, and we'd have A tails added to them. But we need to add molecules or sequences onto the ends of these DNA fragments in order for them to be sequenced. So adapter ligation is a critical part of the library preparation method. So the NEB Next adapters are 65 nucleotides long. And I'm saying nucleotides because they are single-stranded. So I'm saying nucleotides instead of base pairs because the adapters are single-stranded. So this is the NEB Next adapter. So 65 nucleotides, well, 65 nucleotides, single-stranded DNA molecule. Now, some important aspects of it. This is the 5' end, and this is the 3' end. Well, a G is going to be found at this end, a G nucleotide, guanine nucleotide. A T at this end, a C right before that. And then in the middle, a U. Normally you don't think of U or uracil in DNA, but these are chemically synthesized strands of DNA, and so sure, a company can add a uracil DNTP at this position. And what position is that? That's position number 32. So over here, there's something important. There's going to be 12 nucleotides. And these nucleotides over here are going to be complementary to the 12 nucleotides over here, 12 nucleotide complement over on this side over here. Now, this is where I think it might help for me to color this in so it matches the colors used in the printed notes. Okay, so these sequences over here I'm coloring green, these over here orange, and the U remains uncolored. Now, there's also something unusual between this C and T. So what is there? So there is a phosphorothioate bond instead of a phosphodiester bond. So phosphorothioate, what is that? So essentially, what is a phosphorothioate bond? So, okay, phosphodiester bond. So by now you should be able to write that out. You already have that memorized, right? Phosphodiester. So let's start with the phosphodiester bond or phosphodiester linkage between two nucleotides. And oh, that's a terrible angle on that bond there. So here's a nucleotide. Here's the 3' carbon. Here is our phosphodiester bond that links this nucleotide to another nucleotide. Now, this is our standard phosphodiester bond, typically found between nucleotides in DNA and RNA molecules. Now, the phosphoro, I don't know why they call it phosphoro, but phosphorothioate bond is very easy to understand if you understand this one right here. What we do is just replace that O by an S for sulfur. Okay, now why would a company want to use this type of bond? Well, our exonucleases, our standard exonucleases, and I am not sure DNA polymerase 1 with its exonuclease activities is an example, but many exonucleases do not work. They process phosphodiester bonds, but they don't process phosphorothioate bonds. So if you want to make a DNA molecule resistant to many types of exonucleases, well, you can use a phosphorothioate bond instead of a phosphodiester bond. Okay, so what was all that for? Well, because there is a phosphorothioate bond right here. And we're going to see this type of bond like several times throughout the semester. So, okay, now we know what phosphorothioate bonds is. We have an overall idea of what this NEB next adapter sequence looks like. So these complementary nucleotides are important. And so I cheated a little bit, and I have this already diagrammed for us. So when this is in solution, what happens is because of the complementary base pairs, right, between those 12 nucleotides at the 5' end and the 12 nucleotides at the 3' end, we get this structure that looks like this, right? So these, we have this little stem right here and this little hairpin, actually a long hairpin right here. But yeah, so the adapters will form this secondary structure with hydrogen bonds forming between these complementary nucleotides here. And look at this, we have a T-tail at the 3' end. So our molecules in the library, our molecules that we want to sequence, all have A tails. Well, this has a T-tail at the 3' end. And this is where the phosphorothioate bond is. Here's our uracil over here. Now, okay, so that's the actual sequence of the NEB next adapter, and it adopts this structure when it's in solution. So what we need to do now, we would need to mix that adapter. Here, let me diagram a couple of these right here. So here's an adapter with a little T overhang, and let's take one of our molecules that represents the millions we have in our solution of N-repaired and A-tailed DNA fragments. Now, what we need to do, or what we want, is we want all of these adapters, we want one adapter stuck to each end of this molecule. So here we have T's on the 3' ends of the adapters and A's on the 3' ends of the DNA fragments in the library. And so what we're going to do is add DNA ligase. So T4 DNA ligase. And T4 is coming up a lot. This is a bacteriophage, and one of my goals this semester is for one of those to be determined classes is cover the T4 bacteriophage. So, but we'll talk more about that later. So T4 DNA ligase, we're going to add that with the adapters and our DNA molecules. And T4 DNA ligase, what it's going to do is it's going to ligate the adapters to the ends of our molecules. So we should end up with something like this, with all of our molecules that we want to sequence looking like this. And you know what? I can color this in. And by coloring it in, it'll help remind me that I forgot to put the uracils in there, didn't I? So all of our molecules should look like this at this point. Then we are going to add an enzyme mix that is called user. I think that's what the New England Biolabs calls it, but essentially it's just a mix of two enzymes, UDG and endonuclease 8. So UDG stands for uracil DNA glycosylase. Oh, and these are enzymes that come up also a couple more times throughout the semester. So this is kind of why we're going over these enzymes. Every enzyme we've heard so far, well, these are very important enzymes for molecular biology. We're going to see them many times throughout the semester for various different things. This one right here, these two are important in DNA repair processes. So what happens when we add these? So essentially what happens is it cuts out the U, and it breaks this bond right here, breaks the phosphodiester linkages here. And so what we get is all of our molecules looking like this. So that's our A right there. So the U's are gone. And, oh, I got a little too messy here, sorry. So we have some bonds here, right? But these, so these are the adapter molecules, right? The green and the orange. They're stuck together right here, but they're not stuck together here anymore. They're not connected anymore, and they don't anneal because they are different sequences. And that's going to be important. That's going to be important for the next steps. So all of our molecules should look something like this. And remember, the sequences between the adapters are 400 to 600 base pair fragments from throughout the genome that we're trying to sequence.